Okay, good morning, everyone. Welcome to our 10 o'clock briefing today, and happy Valentine's Day. Great day to talk about depression and aging. 10 o'clock briefing is Depression Accelerates Aging in the Brain. And our speakers today are Irina Esterlis, Associate Professor of Psychiatry at Yale University, Etienne Sibyl, Professor of Psychiatry, Pharmacology, and Toxicology at the University of Toronto, and Daniela Kaufer, Professor of Neuroscience and Integrative Biology at the University of California, Berkeley. So we will get started in a moment. I just wanted to remind you that we will end the event at about 10:45 so we can prepare for the next briefing. But if questions go beyond, we will speakers will be available in a follow-up room. So without further ado, Irina Esterlis.
Thank you, and thank you all for coming here, and of course, thank you for AAAS for choosing our symposium for this exciting media briefing. And I am actually going to move from my slide presentation to the podium because I think it's easier for you guys to see that way. So, do I have to still press this? No. Can you guys hear me? Okay. So in healthy brain, what we think, you cannot even see the laser pointer. Okay, so, yeah. In healthy brain, we have synaptic homeostasis, which is on your left. So the number of synapses, dendrites, and size of boutons are normal. In the middle, we have aging. This is a slow process. It doesn't happen overnight. And the number of boutons is going to be smaller, the dendritic spines are shorter, and the number of synapses are going to be less. And this is proceeds over many decades, and you're not going to notice cognitive, emotional, or social changes until later in life. On the right, on your right, we have depression. So we think the changes that are associated with aging in terms of synaptic density are even greater in depression. When we combine the two, we're going to have much more robust changes that are associated with cognitive and emotional and social decline. And so here we did a large study. So I do positron emission tomography, and we did a large study in individuals who are healthy controls as well as those who are depressed. So in gray, you have individuals with depression, individuals who are healthy controls. In green, we have individuals with depression who have low severity depression, and in blue on the graph, you have individuals with depression who have high severity depression. And as you can see, those with high severity depression have lower synaptic density as compared to those who have low severity depression or healthy controls. And this is in the prefrontal cortical regions in the hippocampus. And on the right, we have a parametric image. So this is how we depict PET scans. The top row are healthy controls. The middle row is the low severity group. You can see visually that they have pretty much the same number of red, orange, yellow spots. These are the hot spots. So to speak, they represent higher synaptic density. And on the bottom row, we have individuals with high severity depression. You can visually see that they have lower synaptic density as compared to the other two groups. So then we decided that we wanted to see the combination of aging and depression and what it does to synaptic density in the brain. The green line you see is healthy controls. We have about a 2 to 3%, I don't want to say decrease because it's not a longitudinal study, but difference in synaptic density per decade is about 2 to 3% in healthy controls, starting with higher in the 20s, 30s, and lower in the 60s, 70s. In depression, however, this difference is about 10 to 15%, so the slope is much more different. It's steeper, it's different from healthy controls, and the change per decade is about 10 to 15% depending on the brain region. And so what does this change mean? So in depression, the lower synaptic density the individual has, the lower their attentional skills, visual, perceptual, verbal memory, and working memory skills. And also individuals with lower synaptic density have higher levels of anhedonia. This is in the depressed aging cohort. And that's all I have. Thank you.
Thank you. Could you bring the next slides, please. So again, thank you very much for the invitation. It's great to be here. Thanks, Irina, for organizing this symposium. And so as soon as the slides will, I can start anywhere, but if you can bring them. So again, I'm in Toronto. I'm also at the Center for Addiction and Mental Health. So a very quick summary of what I'm going to talk about tomorrow, and starting with a clinical perspective. And so we all know about this categorical definition of disease, such as depression, schizophrenia, Alzheimer. But if we look at symptoms, cognitive symptoms, which include problem with working memory, decision-making, planning, etc., actually core symptoms of many neuropsychiatric disorders. They're highly prevalent. They define disease of old age. They persist in remission. They actually predict relapse in the context of depression. And it's thought to be really that they're the core of some of the disease. But interestingly, they're really not treated by any current medication. So whether antidepressant, antipsychotics will affect some symptoms, but really they don't touch the core of the cognitive symptoms. So and this is a little bit in the context of the fact in neuropsychiatry there hasn't been new drugs, at least drugs with novel mechanism of action for at least 60 years. They've discovered what is in the market has been discovered by chance, and basically farmers have tweaked those molecules but haven't come with new mechanism. So what we really need is novel drugs with novel mechanisms. So how do we do that? Over the past, I would say 15 years, we've gone back to the human postmortem brain, did a lot of what's called, those omics approach, large scale, looking at 20,000 genes, etc. And to make a long story short, those studies are now pointing to a specific deficit in a cell type. And what you see on the right here, it's a canonical microcircuit, different cell types that regulate each other and that are really essential for information coding in cortical layers. So really a basic function for cognition. And what I'm showing on top, those SST, which stand for somatostatin, it's a molecular marker for the cell type that regulate input. So it's a very specific function. We've shown now that those cells are specifically vulnerable. We showed that in the human brain, and then went on to rodent using genetic and environmental manipulation, show that these cells are weak link, and they actually cause some of the mood and cognitive symptoms. So based on that, we zoomed in, if you want, on those cells on the left and on the connection, those synapses between cells, and try to identify areas or targets that we could work with with the goal of remediating the pathology. And we've been focusing on this postsynaptic receptor, so the receptor that mediate the function of those cells. The name of this receptor, so those cells are GABAergic. This is the main inhibitory neurotransmitter in the brain. And a specific subtype is called alpha-5 GABA-A receptor. So the point is that it's very specific in terms of cellular localization, brain area. And over the past 5-6 years, we work with medicinal chemists to develop small molecules that bind and actually increase the function of this receptor. Again, with the hypothesis that this would not just potentially have an effect on symptoms, but also target the pathology. And what I'm showing you here in mice, using a Y maze on the left, which is basically a test for spatial working memory. You can train a mouse. Within a week, the mouse will perform at 80 to 90% accuracy, knowing which arm it visited previously. You can stress a mouse for a week, as shown on the upper right, and you see in white, the performance go down to close to 50%, which is basically by chance. So the working memory is not working anymore. And what I'm showing you on the right is one example of dose-dependent rescue of those phenotypes. So within 30 minutes of injection, either injection or through water, the mice basically perform as control mice. And we've reproduced that. It works in male, female, over 15 times. What is exciting also is when we looked at old mice, without any stress, old mice will have deficit in working memory. And here, after 30 minutes after an injection, after daily injection for a week, or even after 2 months, we show that we can basically have old mice perform to levels very comparable to that of young mice. So these are exciting. The new data that I'm showing you today for the first time is when we look at the brain, so similar to what Irina just showed you, with age and stress, those cells tend to shrink. You lose synapse, you lose dendrites, and now we're showing, so this would, oops, sorry about that. Now, okay, there, showing you on the left, you can see that with age, or you can see on the graphs on the right, the synapse number go down, and now we show that 2 months of treatment, we actually bring those levels back to level of young mice, so somehow rejuvenating the brain, if you like. And this is just a quick summary. I show you, age and brain disorder affect this particular cellular component, and targeting it as both symptomatic and what's called disease-modifying potential. So we now moving forward and hope to start testing this compound in a clinical setting in approximately 2 years. Thank you.
And we were able to ask a lot of questions about now the mechanism. So what exactly is happening whence this is coming in. And it was really interesting because turns out that there's a very specific mechanism. This protein gets into a specific cell type called astrocytes, and that was surprising because you think of neural function, you think of neurons, but it went into a different cell type, and it binds a specific receptor, and it activates this neuroinflammatory response. So there's a lot of inflammation now in the brain, and this inflammation causes a lot of pathological signaling cascade, and changes in neural firing, and changes in the way that the brain functions and different cells talk to each other, and a lot of it has to do with inhibitory and excitation cells, so the GABA cells that you just heard about. And finally, that leads to neural dysfunction and cognitive impairment. And then we asked ourselves whether this is something that is really causing that or just something that correlates with that. And so we took a young mouse, and we took this protein and infused it into their brain. We put a pump in, we infused that protein, and that was enough to recapitulate aging in that young mouse. So it looked like an aged brain when we probed it on different ways. We also did the opposite. We did a genetic knockout of this receptor. We specifically took out this receptor from those cells and show that we can block this brain aging. So that got us really excited. And we decided to pair up with a medicinal chemist and create a small molecule inhibitor, a drug, if you will, that will have very good clinical properties. And we tried what happens when we put this drug into the animals. And that was the most surprising and really exciting part of it, is that we were expecting that we would maybe be able to prevent future things, but what happened is we were able to reverse. And so if you took an aged mice and you gave them this drug, we were able to reverse the network dysfunction that you see here, that is EEG recordings of those animals. We were able to reverse the molecular phenotype of aging in that brain. We were able to reverse vulnerability to induce seizure, this hyperexcitability that we see in the cells. And most importantly, we were able to reverse the cognitive phenotype such that those aged animals now performed at a young animal level. So this is where we are now. This understanding from the mice is very exciting for us. The next step is to ask those questions in human brains and to work on the development. And those are the two graduate students in my lab, Vlad Sinatorov and Aaron Friedman, who just recently graduated. And this is the new space that UC Berkeley put in for an incubator, and we got some grants to try to develop that towards a translational medicine. Thank you.
Oh, wonderful. Okay. Thank you. That was uplifting at the end and all. Thank you to all the speakers. So we'll take questions now. We have a microphone that we can bring to you. If you'd like to ask a question, please raise your hand. We will bring it over, and please state your name and affiliation before your question. So we'll start with two here in the front row. Thank you to our mic runner.
Hi, I'm Vijay Shankar. I'm a freelance science journalist. My question is to the whole panel here. So part of your experiment was to test using a small molecule drug candidate, if I can say that. So for how long it was necessary that the small molecule had to be administered? Or if you administered once or twice or maybe thrice and then you just leave the mice and then the brain aging reverses? Or, in human terms, if it were my grandma or grandpa who has to take this future drug, should she or he continue taking this drug? Or is it okay just taking once or twice or thrice and then altering other aspects? Because aging is, also socio-cultural and food and everything else. So, yeah, I was wondering if you could answer that.
Yeah, I was hoping it was still up, but you could see the timeline, and it was a bit of a surprise for us. So you start seeing effects within 24 hours of the drug, which seems to be too good to be true. So we went down to the molecular level and saw that we're already seeing a bit of a change in 24 hours. For better effects, we used 7 days of the drug as the minimal effect, and then you see the cognitive effects. There's also a continuous effect in the washout period. So in my slide, you could see that we then stopped the drug for another 7 days, which follows a clinical trial protocol that we might want to use in the future, and you still see the effect during the washout.
it's clear that on a chronic level, you're going to have to administer it repeatedly, but maybe in bouts and not constantly. One of the things that we're excited about is that there's not just the drug aspect of it, but there's a biomarker aspect of it. And because we have now the techniques to look at subtle changes in the blood-brain barrier, I would say, you could combine that with a biomarker where you're screening and saying, this is when the blood-brain barrier is open, this is what I need to do. Or you could imagine screening middle-aged people and say, this is a person that is now at risk, we should treat them. And then you can follow with a biomarker.
I could tell you in our case, because we have small molecules also, so the mechanism is different in our case. They work after 30 minutes, basically the time it takes to reach optimal level in the brain. And because the acute effect is to restore the neurophysiological function of those neurons. So behaviorally, we see an immediate effect. In the context of our drug, actually, as important question is, will the effect last when we repeatedly administer? Because it could be a problem with some of the type of target we have, and we show that weekly, daily exposure for a week works. For up to 2 months, it works. And we know that now at least 2 months, it takes longer if you want to restore the structure of the neurons. We know it happens at 2 months. We're going to look at earlier time point. So behaviorally, acute effect is prolonged. Structurally, takes weeks to. And the next question is, if we stop now, how long does it stay at that younger level, for example, or do we have to continue giving the drug to, in this case, to our preclinical models?
I guess the one thing I would like to add is just to be mindful and careful of looking at animal and cell studies and then translating to human. So in animal work, as related to my work, administration of ketamine, which is an NMD antagonist, reverses synaptic loss in models of depression in animals within 24 hours, and the animals now look like control animals and they behave like control animals. But administration of one dose of ketamine to a human, to some humans, it will reverse depression for a few days to a couple of weeks, and then some will not. So I think we just have to be careful when we ask these questions and whether we want to give it to our grandmas and grandpas. We don't know yet. It looks like positive results, and so we're very excited. And so we would like to continue doing this work and investigate this.
Hi, this is Purti, and I am a science writer and reporter for a science media portal in India called Research Matters. And my question is a little bit on a broader sense, that when you say aging, so we did hear Irina talk about, synaptic, defects in synaptic activities that goes on with age, and then there is this blood barrier thing that opens up when you age. And so when you talk about drugs to treat that, does it address all of these things, or is it one specific thing which might have impacts on the other? How do you say, these drugs actually reverse aging in that sense?
Yeah, I could try. So I think, you're right. Aging is a lot of different things, right? And it's going to entail a lot of different things. One of the things that we were excited about is sort of figuring out that there is a masking of the of the proper neurotransmission, right? And so there's neurotransmission, and that goes wrong in aging. And neurotransmission will involve the GABA system, and it will involve the synaptic, and we are talking about something that sits sort of on top of that and affects them. And so there are going to be a lot of different mechanisms that are involved in that, and a lot of different ends to that, right? So one way would be to inhibit our receptor, another would be to inhibit the GABA receptor, another would be to change the synaptic density. There's definitely a lot of things, but each one of those sort of gives you a piece of the puzzle to understand what happens in the brain.
Yeah, I would add to that we actually all age differently. And different mechanisms may be engaged in different people, and there's such a thing as successful aging, and And there's one interesting aspect of the novel treatment we're developing is they have no effect in control subjects. So if there's no deficit, they don't do anything. So it's not like they're going to make somebody like a genius or whatever. But if there is a deficit, and again, in some, for example, in the context of depression or aging, some people may have those deficits, some not. So the idea is that we need to use biomarkers, as we mentioned earlier, and we're developing those to identify who specifically has this particular pathology, and those are the people we would want to treat.
Questions, 2 questions in this row.
Clive Cookson from the Financial Times. I wanted to ask about the patent and intellectual property position of the drugs that at least 2 of your groups are developing, and whether you're collaborating with or have spun out any companies.
Yeah, so we have a couple of patents. We have a patent on the new ligands, on the use. Actually, the last slide I showed you where we, I mean, I'm simplifying, but when we regrow those neuron cells, we actually put the patent in yesterday, the application, so that I could talk about it at this meeting. So yes, it is protected. In our case, I'm putting a lot of the foundation money that I got by moving to Toronto into this project. We have a commercialization development grant from the CIHR, Canadian Institute of Health Research, and we're in discussion with VCs. We're trying to bring the whole project. We think a year away from submitting an investigation, new drug application, a year and a half. So we have some funding secure, but we're talking with venture capitalists, we're exploring all avenues.
We've filed through Berkeley an IP for the use method, and we're in the process of getting the actual molecules as well. And we got a really fun grant actually from Berkeley called the Baker Fellowship. So it's an entrepreneurship fellowship that walks us through towards commercialization, and we're doing it right now and talking to VCs and so on.
Question, Robin.
I'm Robin Williams from the Australian Broadcasting Corporation, and I'm also extremely old. And I'm a hedonist. And I'm wondering whether there are any exercises without the drugs that you mentioned that might have an effect possibly on the synapses, less likely maybe on the blood-brain barrier, that improve. And I'm thinking quite seriously of activities like exercise that seem to ameliorate depression to some extent and certainly improve memory, things that myself and Clive Cookson can look at before you have the drugs available generally.
So in human, nobody has really looked at effects of anything on synaptic density because we just developed this marker a couple of years ago with the Yale Pet Center, so it's in vivo prime time for us. But from everything that I have read and learned, I run, I lift, and I do Sudoku, and I do science. So those are my recommendations to my family and to anybody who's asking.
Yeah, no, I mean, exercise, mental exercise, physical exercise is known, has effect, it's been tested physically, but some of the biological mediators have been identified. So you may have heard of, brain-derived neurotrophic factor, BDNF, is induced by both types of exercise, and they have broad effect, those factors, and they're actually upstream. We've published quite a lot of work on showing that BDNF is an upstream factor responsible in large part for the deficit we see. But so targeting that with a pill is much more difficult because it's broad ranging. So that means side effects. So it's much better to. So the answer is, exercise works, but people prefer to put it in a pill. That's the point.
Question over here, and then we'll go back there. Yeah.
I'm Nuala Moran from BioWorld. I wonder if the three of you could reflect on how important the BRAIN initiative has been in establishing and developing the tools and techniques which have obviously been so important in the advances that you are all making.
I'm a big fan of the BRAIN initiative, and we benefit a lot when we first saw, so I'm going to make 2 points. When we first saw deficit in this marker 10 years ago, very little was known about those cells. 10 years later, I mean, we can look in the literature. Every week, there are new findings due to new technology, due to money that is put in basic research. So there's a tremendous amount of knowledge, and I feel like I'm standing on the shoulder of all those giants. And I like to compare sometime to what's happening now in neuroscience, psychiatry, et cetera, to what happened in cancer. You know, when Nixon declared the war on cancer, I think people very quickly realized that this was great, but they just did not have enough knowledge to make a serious impact rapidly. And it took a long time to gather that knowledge. And I think what the BRAIN Initiative has done is reverse that. You know, when Obama started the initiative, you know, over 10 years ago, we benefit now for this tremendous amount of new knowledge that allows us to study disease into a different way, greater depth, new tools. So it's been incredible help for us.
Anyone else want to comment on that? Okay. Okay, great. Well said. All right, we had a question over here, I think.
Hi, I'm Elise Zhen from China, freelancing reporter and this year's York Alert Fellow. And my question is, especially I want to ask Ms. Estelis about the social implication of how the depression can worsen aging. So is there something that would change the society, how we view the aging as a social phenomenon?
That's an interesting question. So you're basically, I'm trying to like rework it in my brain. You, the effect of depression on the aging and how that will affect social, how the society is going to perceive aging or depression, because they're both perceived quite poorly.
What would be the implication if we could understand, like better understanding mechanisms and or to do something to help the elders to, yeah.
So I'll go a long way a little bit. Last year in JAMA Neurology, there was a paper published by the Yale group. Chris Van Dyke is the senior author. He does a lot of Alzheimer's research. And they used the same tracer that I just showed you today to study aging population of healthy controls, individuals with mild cognitive impairment, and individuals with Alzheimer's disease. And they saw that the hippocampus was the area that was suffering the most from cognitive changes, or associated the most with the cognitive changes in basically Alzheimer's disease. And so looking at that and looking at depression and what we're seeing with depression in the small sample that I showed you today, that around 40 years of age is when we can start differentiating individuals with depression from controls in their cellular structure. If we can identify those who have lower synaptic density, for example, in the hippocampus, and we can identify them early on, and we can treat them with whatever medication that right now we likely do not have yet but are being developed and tested, if we can induce synaptogenesis, reduce cognitive decline, restore occupational functioning and thus social functioning, I think we can prevent or delay, probably at least delay for now, a lot of the dementia disorders, related disorders, and suffering associated with depression, and restore social and occupational functioning to these individuals so they can live longer lives. Does that answer your question? Yeah.
You mentioned some mental exercise in the form of Sudoku that might be helpful in this context. Are there some activities that you would say are definitively like antithetical to that, like being on the cellular phone? Are they unhelpful in that way? Has anything been found so?
I am not sure, but I think mindless things are not as good for brain development. On the other hand, I think it is important to relax and let yourself enjoy some mindless activities because you can't be on all the time, and so you need to restore. Because I think if you keep stressing about having to exercise, having to mental exercise, having to read for synaptogenesis or whatnot, then it's going to reverse because now you're going to be stressed, and so we saw what stress does.
Great. Okay. If there are no other questions, that concludes our briefing. Thanks so much to our speakers. We really appreciate your time and insights. And I believe Irina and Daniela could be available for follow-up questions afterwards if you have any others. And there is a room on this floor, the Buchanan room, for that. Thank you.